So welcome back all. So I'm Justin Nono from the Ministry of Scientific Research here in Cameroon and also at the University of Cape Town where I do lead the research group. I will moderate this session with Dr. Anos Boufras and some very recent finding and innovating research on the schistosomiasis. So the first part then of this session 2 will showcase 4 different talks touching on morbidity innovative tool, vaccines, modeling, and biocontrol. So the format is 10 minutes talk, followed by 5 minutes then a Q&A for each speaker. As in the previous session, you will just dive your question in the Q&A then panel. So our first speaker then of today talk will be Dr. Odiere, Maurice Odiere from the Kenya Medical Research Institute. And he will then talk to us about the use of a tablet-based system to perform abdominal ultrasounds in a field investigation of schistosomiasis-related morbidity in Western Kenya. So Dr. Odiere, the floor is yours.
It's my pleasure to be part of this meeting and an appreciation to the organizers of the GSF for inviting us to this meeting. I wish to share with you the results of a pilot study on the use of a tablet-based system for conducting abdominal ultrasounds in a field evaluation of schistosomiasis-related morbidity in Western Kenya. Now, the MORBID study, the MORBID is an acronym. It stands for Morbidity Operational Research for Bilharziosis Implementation Decisions. And it was launched in 2019. The goal of this study was to identify better methods to detect morbidity for schistosomiasis control program, basically looking at the correlates of morbidity for infection. As we know, morbidity is usually assessed by several methods, which including among them ultrasound of the liver and spleen. Now, for this study, we targeted an area of Western Kenya where Schistosoma mansoni is endemic. We worked in 45 villages, targeting approximately 200 residents in each village, which included preschool-age children, school-age children, adolescents, and adults. Now, ultrasound evaluation has been used or recommended for use in schistosomiasis, in assessment of schistosomiasis morbidity, because it allows for direct visualization of the liver and spleen, but it also has other inherent advantages that include among them the fact that it's non-invasive, it's relatively easy or simple to perform. It generally has a high community acceptance, and then a quick turnaround in the results that are expected. But importantly, it has the ability to distinguish between fibrosis that's specific to mansoni as opposed to other causes of hepatic disease. Now, unfortunately, ultrasound, the utility of ultrasound has not gained so much traction in the field due to several reasons. One of them is the cost of machines that are quite high. The fact that the older models that were used were quite bulky, large, heavy. They required a generator and fuel to be used in the field, so there are logistical challenges. And then there was also lack of capacity in terms of the expertise to perform ultrasounds in the field and to interpret the results. Luckily for us, we have had the opportunity and experience to use or to conduct ultrasounds during several of our studies, key among them the SCORE study that most of you might be familiar with. For the SCORE studies, the machine that we used was a refurbished Aloka SSD 900 machine. It weighed roughly 15 kilograms. Of course, it required a generator and a fuel, but it could not store images electronically, so we had to purchase a printer and special thermal paper. One of the downsides with the thermal paper is that the images that were printed faded quickly. And then there was another significant challenge in terms of the cost. Around 2011, that cost was about 11,000 USD. Currently, I think those machines cost anything between 22,000 to 36,000 USD. And as a sample of the Aloka machine is an image of a sample of the machine that we used is shown on the right-hand panel. So fortunately, now we have the tablet-based ultrasound systems that have entered into play. This basically consists of a transducer that's connected to a compatible smartphone device. And they're ideal for field applications for several reasons, including the fact that they are light in weight, so they are easy to transport. They do not require an external power source during the period of conducting the exams. They have excellent resolutions, that if you compare that with the clinical machines. And then they allow for capturing of high definition images and short videos that can be saved for review at a later time point. For the MORBID study specifically, we used the Philips Medical Imaging Lumify system with specific broadband, and it's accompanied transducer, and also a compatible Android tablet. Luckily, or the other advantage with this system is that the transducers can actually be leased, which would ultimately reduce the cost. In our case, we actually leased 4 transducers for a period of 6 months. We extended that slightly due to a delay in completing the study activities. And the total cost for that was about $1,200 US. We purchased the tablets locally at a cost of about $140 USD for each of the tablets from the Philips company that's located in Nairobi. And the system consists of a tablet, as you can see on the right-hand panel, and also the transducer that's attached to that tablet. Now, from using these tablets in the field, we're able to observe several things. The battery life was sufficient to perform about 25 ultrasound examinations in a single day. We're able to store the images electronically, and then at the end of the day, we're able to recharge those tablets and then download the images on external hard drive. We were able to then use those images to conduct our scoring for hepatosplenic pathology based on the WHO recommended NAMI protocol. And we did a total of 6,483 ultrasounds, abdominal ultrasounds. One of the other challenges that I had mentioned earlier is the fact that there's lack of capacity in most of the settings. So now in our case, we used an innovative approach where we hired recent graduates of medical diagnostic imaging program in one of our local universities who are training to be sonographers. Now, these graduate students were then sent to for an intensive 3-day training to learn about schistosomiasis-specific ultrasonography at the Kabatarine Schistosomiasis Research Center in Uganda. I'd just like to share a few slides with you on some of the results. The first one, of course, is we had identified 2 areas where we wanted to conduct the study. One was the endemic region, which is Siaya, where we had a prevalence of mansoni of about 25%. And then the control area was Vihiga, which is about 65 miles away from Siaya, where we recorded a prevalence of about 1% for mansoni. So that kind of made it a good control area for our study. The specific prevalences for each of the age groups is indicated in the table. Now, in regards to the liver image patterns, we were able to pick or observe image pattern from A up to D, with the exception of pattern F, which is usually the more chronic form. The liver pattern A usually represents the normal liver pattern, as you can see in the panel. Panel B is the starry sky, or it looks like stars in the sky, as you can see. Panel C is called the rings and pipe stems. The echogenic features look like rings and pipe stems. And then pattern D is the rough, looks like the hairs on the head or neck of a bird. And then pattern E is usually what we refer to as the patches. So pattern B usually represents the intermediate transition stage from the normal to significant pathology. And then in terms of the other pathologies that we're able to assess, we observed an age-dependent effect of infection, of Schistosoma mansoni infection with regards to pathology, where we noted that pathology increased with age. With adults, for instance, chances of having periportal fibrosis was 6 times higher in adults compared to adolescents who had Schistosoma mansoni infection. And we also noted the same trend in advanced periportal fibrosis and the other conditions. We also had an evaluation of the organ sizes or organomegaly. And as expected, we observed a significantly higher proportion of organomegaly in Siaya, which was the endemic area relative to Vihiga County, as you can see in the table, with the exception of splenomegaly. Now, some of the community benefits, what I'd say they're, beyond the negative effects, direct effects that we're able to see, health effects that we're able to record with regards to schistosomas and infection. We also observed other non-schistosomiasis pathologies, for instance, uterine fibrosis, PKD, hydronephrosis, fatty liver, liver cysts, and importantly, another congenital condition called situs inversus, which is a condition in which the major visceral organs are reversed or mirrored from their normal positions, as you can see in the panel on the right. That we observed in just one participant. And then the other, what I would call social benefits that we're able to observe in our study is we're able to detect pregnancies. In fact, some of the women were not even aware that they were pregnant. And, just beyond detecting the pregnancy, we're able to actually determine the gestational age and the expected date of delivery, and also the gender of the fetus. Now, in conclusion, I think our study contributes or adds to the body of literature that shows the utility of ultrasonography in assessing schistosomiasis-related morbidity. But in addition, we've been able to capture other ancillary community benefits, as I indicated, diagnosis of pregnancies, the diagnosis of the other non-schistosomiasis pathologies. And we also are excited at the prospects that the advances in technology currently, are making the ultrasounds more portable and less expensive. And this will likely make it more feasible to incorporate into country control programs in the not very distant future. And this, I think, will be very important because integration of this into control programs can actually help countries in not only reducing morbidity, but also in achieving WHO's vision of universal health coverage by providing communities with access to advanced health services. I'd like to register my appreciation to the entire Kenya Morbid study team, the task force for global health, and all the study participants. Thank you.
Well, great talk, Dr. Odiere. It's quite interesting then here, then first of all, to see how diverse and counterintuitive then the onset of this periportal fibrosis was within the adolescent population. And I'm sure most then will relate to how then useful this will be when you had, for example, getting to the field and to carry a whole generator and fuel as per then the whole proceedings to make sure that the imaging then analysis were carried out then without interruption. So that's a great addition. We do have a couple of questions actually from the Q&A. I will first take the one from Ebube Odoya, who is asking about training in Nigeria. So the training of the clinicians. So how that could be made then feasible. Dr. Odiere?
Okay, if we could just ask you to pop your video on, please, Doctor, and then we can spotlight you so that you can answer the questions.
Is he with us, Dr. Odiere?
Yes, he is with us at the moment. I can see he's in the room as a panelist, but his video is not on. So if you'd like to join us, perhaps you could pop your video and your mic on. Maybe we're having difficult connectivity issues. Hi, Anoop, would you like to?
I was just going to say that perhaps Maurice might be having some internet issues at the moment.
Yeah, I just read from him that he cannot see the video options. Can you see can you hear us actually? And could you answer even the audio feed if it's working?
Yeah, if you pop your microphone on, if you look on the left-hand side of the bottom Zoom menus, and press the unmute button.
Hi everyone, sorry for that. So I've been able to join using the panelist option for the question. So with regards to training, I think what is required is basically the standardized schistosomiasis specific ultrasonography training. And I think there are different activities or opportunities for this to happen. So if there are clinicians, for instance, in a particular country that would wish to join or participate in some of these, that is something that can be arranged, especially with the standard training centers that have been established. So for us, in our case, for instance, the opportunity to do that at the training center at Bugoigo in Uganda was really timely just before we started the study. But I totally agree that getting clinicians on board just to increase also the index of suspicion for some of these diseases, I think is definitely a good way to proceed.
Great, Dr. Odiere. So we do have Bonnie Webster that wants then to ask a live question. Bonnie, can you then on your camera as well as unmute to ask the question directly to Dr. Odiere?
Hi, thank you very much. So my question is related to how acceptable the procedures are in the communities.
So that's a very interesting one, Bonnie. I think interestingly, what we learned from our setting here is that actually they are very, we were very well received. Typically, I think some of the community members especially find some of these procedures to be a little bit too clinical, so they don't get to access some of these activities out there in the community. So this was kind of like taking the clinical procedure and taking it to their community. So the reception was quite good. Of course, we had to take into consideration some sensitivities in terms of creating isolated areas where we can conduct the exams, making sure that we have the right ultrasonographers conducting exams, gender issues and all that. But in general, I would say it was very well accepted. In fact, the members, I think, appreciate the fact that they had missed out on some of these things because of issues of affordability and access. So yes, I would say definitely very well received.
Okay, thanks, Dr. Odiere. I do see that there are quite a lot of questions that could be redirected to Dr. Odiere since we are running them beyond schedule. And Dr. Odiere will then provide those to us, and those will be uploaded then with the recording on the GSA website. So thank you very much again once more, Dr. Odiere. Very wonderful job there, actually. Very innovative too. So,
and sorry, Justin, just to quickly interrupt and say, Dr. Odiere, if you would like to type in an answer as well to these questions, you can do so on the Q&A panel. But yes, we can also send them to you and we can upload them onto the website. Thank you, Justin. Apologies.
Sure. Thank you, Anouk, for that compliment. So I will now introduce our second speaker. That is Mrs. Driciru. She's from the Uganda Virus Research Institute in Entebbe, and she will then talk to us about the immunological consideration for schistosoma vaccine development, transition into endemic setting. So Mrs. Driciru, the floor is yours.
Thank you to the team at GSA for this opportunity. I am Emmanuella from LSHTM Uganda, and my presentation will be about the immunological considerations for schistosoma vaccine testing, transitioning to endemic settings. Let me just share my screen. And this was published as a review paper in the Frontiers Immunology earlier this year. So as we know, schistosomiasis is still a significant public health burden in low-income countries, mainly due to the heavy dependence on lake waters that are heavily infested with schistosome transmitting snails, coupled with inadequate sanitary facilities. And Praziquantel-based mass drug administration has not been sufficient to achieve elimination targets, mainly because Praziquantel does not prevent reinfection, and thus the need for vaccine whose development has been hampered by several factors, ranging from host to environmental to immune to parasite-related factors such as the complex antigenic switches between the different groups during the life cycle development. Nevertheless, progress has been made from the use of whole live attenuated saccharine to the more advanced antigen-specific approach that's more applicable for human use. So we currently have 4 vaccines, candidate vaccines, transitioning through the different phases of human trials. So as these vaccines move to trials in endemic settings, it's important for us to give thoughts to the challenges, immunological challenges that these trials may face in endemic settings. And one of these is a pre-existing infection, whereby we see exposure happening as early as in utero or during early childhood stages. We see B and T cell sensitization already occurring even in newborns, and cord blood exhibiting worm-specific IgE and cytokine responses, as we can see in this Kenyan infant and maternal cohort compared to the non-endemic North American cohort. And we also have development of natural acquired immunity that happens with age-dependent cumulative exposure, and all this may conjointly play a role to impact vaccine development, and this is already a negative one for BCG vaccine. Luckily enough, it's not the case with the recombinant SM14, whereby we still have we still have good responses despite prior exposure. And this agrees with animal studies that show that successful vaccination is still possible in animals that have pro-infection. And this suggests that schistosoma vaccines may actually retain immunogenicity despite pro-exposure, but this impact may vary with antigen. Another factor is differences in baseline immune profile. Compared to Caucasian cohorts, African cohorts tend to exhibit an activated immune microenvironment comprising of highly differentiated B and G cell repertoire, pro-inflammatory monocytes, activated and exhausted cell repertoires, such as we see here in this Ugandan cohort that is showing exhausted and activated NK cells, NK cell populations compared to their Swiss counterparts. And such a state is associated with a negative specific antibody development induction, and yellow fever, BCG, and MVA responses have already been shown to be poor in African infant cohort compared to their UK infant cohorts as a result of these factors. And this partly stems from genetic differences, especially the HLA and cytokine gene polymorphisms, as seen with the vaccine heterogeneity, with the measles vaccine heterogeneity. And therefore, booster doses, use of potent adjuvants and higher antigen doses may be important, fundamental for better responses among endemic populations. Co-infections are another factor, of which the most common is malaria co-endemicity, as we see especially in high schistosome transmission communities. The fishing communities also have rich breeding ground for the malaria vector, and nocturnal fishing activities increasing exposure. And asymptomatic malaria is of most cause for more concern because it's the most common manifestation in the infection peak age and young adults that are the more ideal subjects for schistosoma vaccine trials. And yet we see an accumulation of a typical memory B cell response in asymptomatic malaria. And what this does, how this affects vaccine response is that such a response results in a distorted big load expansion and antibody maturation. And we already see decreased responses to new cellular pertussis and meningococcal vaccines in the presence of asymptomatic malaria co-infection. And thus testing and treating participants before vaccinations is very important, and has already shown improved responses to tetanus. This has already demonstrated improved response to tetanus vaccine. And hence fundamental studies to ascertain the effects of asymptomatic malaria on each vaccine candidate is very fundamental to handle this challenge that may be faced. Soil transmitted helminths that exhibit similar responses as schistosoma are also another factor. And the strong Th2 response and STH-induced immune suppression has a negative implication on vaccination as well, such as BCG vaccine and the oral cholera vaccines. And testing and treating subjects prior to vaccination is very helpful, as we see in this graph here. Albendazole treated subjects show better response to BCG compared to their counterparts that are untreated. And therefore, test and treat is the way to go to handle STHs, as long as there's consideration given for optimal timing of treatment and intensity of infection to prevent possible antigenic competition. So given that MD has been implemented in many endemic morbidities for decades, it's so important for us to look at vaccination in the context of PZQ treatment. Though PZQ is considered non-immunogenic in itself and would have no effect on vaccination, the mechanism of action is what induces several immunological responses that may potentially affect vaccine response. As we see that the tegument shedding unmasks quite a number of antigens that trigger responses and therefore result in better antigen recognition, amplification, alleviation of immune suppression, and even enhanced cytokine response and polarization of cytokine response. And co-administration has been shown to be advantageous to measles vaccine response and GST, and might be the better way to go for endemic settings to achieve a better vaccine responses. And so considerable investment into early phase trials and fundamental studies to try to understand and disentangle the effects of these factors at play is very important, and inclusion of several trial arms to cater for the subpopulations within endemic populations that show distinct immunological and disease patterns. And careful selection of that analysis methods is important for later stage trials, and controlled human infection models are also very fundamental for endemic settings to not only accelerate the vaccine testing pipeline, but also to study these factors in the light of vaccine trials in a cost-effective and time-effective manner, since this model requires a few number of participants and a shorter period of time to investigate immunological and vaccine responses. And the first controlled human infection for schistosomiasis has already been established by the team in Leiden and proven safe and tolerable. And what is left is to transfer this setting to an endemic population. And so selection of ideal trial subjects in endemic settings is a huge task given the several factors at play, but it is fundamental for us to invest in baseline and preparatory studies, or as part of trial protocols, investigations to accurately establish the impacts of these factors and to transfer cheese to endemic settings in order to have appropriate translation of vaccine trials and implementation of vaccinations in endemic settings. Thank you for listening. I acknowledge the following.
Okay, thank you, Mrs. Driciru, for this interesting talk. It is true that it's now more than ever timely for us then to know exactly what all the underlying basis for the differential vaccination, the response that we do experience, especially now when we have for so long neglected primary research or basic research here in endemic settings, where most of the translational undertakings are meant to be applied fully. So this is very timely, and I think is a rightful call to actions. So are there any questions that were asked to Mrs. Driciru? I will then venture to ask one, which is about the need for us to flatten all these underlying conditions so as to better assess the impact of a vaccine. In as much as I do understand the approach, would it not be ultimately better to have then a vaccine preparation that will work on top on all of these background, irrespective of what they are, so as to persist then beyond all these co-infections, previous exposure to treatment, previous exposure then to the parasite, and in so doing then ensure that whatever we will apply then to the field in natural circumstances is going then to circumvent all those limitations. So will it not be better actually to incorporate all those background and make sure that we find something that works on top of these backgrounds?
Thank you for that question. I hope you can hear me well. Well, yes, in the ideal situation, yeah, it would be good to have all these factors, to have a vaccine that suppresses all these factors and have sort of a super vaccine. But we have to acknowledge that these factors do not also play their parts as a bundle in these different populations. As I mentioned, endemic settings also have subpopulations within. We see very diverse immunological differences between urban and rural settings. In endemic settings, we see populations that have had more intense exposure to schistosomiasis, while others have had minimal. So these interplays, they play at different levels, and so that is why we're saying that we can have these vaccines, but if we're going to test them in endemic settings, let's have smaller fundamental studies to look at how each of these factors play, or we can incorporate these fundamental studies as part of the trial studies, as trial arms, to look at how they would affect the vaccine, because some of these factors have either negative or positive impact depending on the antigen type in the vaccine, or depending on the vaccine. Is it a live vaccine? Is it attenuated, or what's the antigen? So there are several factors at play. That's why we're saying, can we consider these several approaches like fundamental studies? Can we consider having booster regimens? Can we consider using different adjuvants and all these modifications? Can we look at all this for each candidate vaccine, so that we don't just conclude that this vaccine is not effective, yet it is, and it's just a factor that is downplaying it? I hope that answers your question.
It does, Mrs. Driciru. So thanks very much. We do have a final, we have questions coming in, but for the sake of time, we will take a final one from Christina. And the other question could be directed to Mrs. Driciru then for the answer in the Q&A panel. So Christina is asking if there are evidences of the impact on malnutrition on vaccination efficiency in this particular context, especially for schistosomiasis vaccines.
So maybe one of the panelists can help me answer that. I personally have not come across studies that would directly affect schistosomiasis vaccine responses, but we've seen several cases with other vaccines whereby malnutrition plays a big role in the responses. Of course, if you're malnourished, your immune responses would ideally be less robust than someone who has been feeding well, someone whose developmental process has not been affected by diets or nutrition. Thank you.
Hi, we appear to have lost Justin for a minute. Are you there, Justin? If you could pop your video on, please.
Yes, actually, I had a slight little internet hiccup here. So very much thank you, Mrs. Driciru, for the talk. And we do have some questions that will be transferred directly into you for answering. Thank you very much again for keeping us in our breath when it comes into the progresses in schistosoma vaccine development. So our next speaker is from the Ethiopian Public Health Institute, the Federal Ministry of Health. This is Mr. Markos Sleshi, and he will talk to us about the use of a geostatistical approach to determine spatial structure of Schistosoma mansoni following 5 rounds of preventive chemotherapy and informing an efficient survey design for national remapping and impact assessment. So the floor is yours then, Mr. Markos.
My name is Markos. I am the survey coordination consultant. I support the Ethiopian reassessment mapping. I now present about using a geostatistical approach to determine spatial structure of Schistosoma mansoni following 5 rounds of preventive chemotherapy. This approach will inform an efficient survey design for national remapping and impact assessment. Schistosoma mansoni is widespread in different parts of the country. Schistosoma hematovium is found mostly in northeastern parts of the country, following Awash River Valley. About 38.3 million is at risk of schistosomiasis, and about 14.6 million school children require treatment. The program conducted 5 rounds of MD for schistosomiasis since 2015. The aim of the geostatistical approach is to develop a national mapping strategy to target MD at sub-district level. Using standard sampling approaches for schistosomiasis to determine prevalence within each sub-IU would not be feasible due to enormous sample sizes, which would cost too much, take too long in multiple phases over several years, require a large, highly skilled workforce. The geostatistical methods need to take into account data previously collected through baseline mapping, impact central sites, and operational research data. Require a more innovative approach. Spatial correlation decreases with increasing distance. These maps show simulations of an area of an average prevalence of 10%. In each of these maps, the spatial structure of prevalence is very different. We decided to use geostatistical methods to model spatial patterns and predict to locations not sampled. This method has the potential for much smaller sample sizes. Random sampling has a limitation as many locations close together will be sampled. Spatially regulated sampling takes into account spatial correlation in data. To design geostatistical surveys, we need to simulate possible distribution of prevalence based on the existing data to determine where to sample. However, after years of MDA, prevalence has likely decreased and may become more focal. First phase in the geostatistical survey approach was to do parameter calibration survey. So we gathered data from selected areas in 2 case studies, SNNP and Tigray regions, to determine the spatial structure of Schistosoma mansoni following 5 rounds of treatment. Data was collected on a much finer scale to generate a gold standard dataset to evaluate survey designs against. We could evaluate potential geostatistical school-based sampling strategies. We then fit PCS data into geostatistical models to get prevalence services. We have made gold standard classification at 10% prevalence threshold to identify areas which needed MDA. We then conducted a number of simulations evaluating the base survey design against this data. To simulate sampling approaches, we first selected the schools to be sampled based on the study design. Then we simulated the number of children would be positive if the 2 children were sampled using the true prevalence surface. Then we fit a geostatistical model to the sample data and calculated the probability of locations being over 10% prevalence. Next, we used this surface to calculate the probability that the Kabale exceeds 10% prevalence and requires MDA. Finally, we compared this assigned treatment category to the gold standard true prevalence surface to compare accuracy. For all survey designs, we repeated these simulations 100 times. To determine optimal sampling design at the national level, we looked at proportion of Kabbales correctly classified from 100 simulations. We compared different sampling designs, all schools, spatially regulated schools, spatially regulated close pairs, and we have seen similar level of accuracy between sampling all schools design and spatially regulated close pair schools. We simulated the distribution of schools across Ethiopia based on population density and school number estimates and identified locations to sample. As outcome, the national sampling strategy was designed to allow for decisions at the Kabbalah level to be made based on a 10% prevalence threshold or 2% prevalence threshold. Spatially regulated close pairs was the optimal sampling design. Number of schools was targeted at 6,732 schools. Additional data will be collected on demographic characteristics, WASH, and other risk factors. Thank you very much for listening.
Okay, thank you very much, Mr. Markos Sleshi, for this interesting talk. I will, there's no present question, so one that, a short one I will lay out there will be about the prediction. Have you tested then already this predictive model so as to see how efficient this is actually in this and redefining the survey design then for this national remapping and impact assessment? Has it been tested already, or is all still then speculative as per your talk?
Thank you so much. Do you hear me?
Yes, I can hear you, Markos.
Okay, thank you so much. So we have tested the simulations against the true prevalence surveys we have created by actually collecting data from the field, from 2 regions. So we have that data, which is the gold standard for testing our simulations against. So, and at the end of the day, after testing 3 different sampling designs I mentioned in one of my slides towards the end of my presentation, so we could able to choose spatially regulated close pairs. And we arrived at target number of total number of schools to have the national data. So we are about to go to data collection. So we are just doing data collection in the coming few weeks. So it's tested, and we have achieved our result for sampling.
That's great, actually, Mr. Markos, and very impressive. I do believe this will be very much helpful for a lot of national control programs in terms of better than rescheduling and then risk defining, then they are remapping, as well as impact assessment at the national and I guess at also sub-regional level. Thank you very much again once more, Mr. Markos, then for your talk. So the next final presentation we will have then for this part 1 of session 2 is from Professor Newmark from the University of Wisconsin-Madison in the USA. And he will talk about biocontrol, basically a rotifer-derived paralytic compound that prevents the transmission of schistosomiasis then to a mammalian host. We do look forward then to seeing that, Professor Newmark. Thank you.
Hi, everybody. Before I get started, I just wanted to thank the organizers for giving me this opportunity to tell you about some of our work to understand how these animals known as rotifers are capable of paralyzing cercaria. Now, as you all know, cercaria have to move in order to infect a mammalian host. I want to show you this movie because I think you'll appreciate it, of what we've done here is postdoc Jay Hunley in the lab has taken a mouse skin explant and stained it with a blue fluorescent dye. Now he's taken a green membrane dye, green fluorescent membrane dye to label cercaria, and now he's going to use a spinning disc confocal microscope to image the penetration of the skin by the cercaria. So now you can see this projection rotating, and now you can see the cercaria interacting with the skin, and in 2 minutes it penetrates it. This will just loop and show you that penetration one more time, as this particular parasite cheated and went through a broken hair follicle. So it took its tail with it, which it usually would not do. Now we're going to talk today about these animals known as rotifers. And it turns out that there are species of rotifers that live as commensals on the host snail shell. This white fringe at the bottom of the snail, if one magnifies it, what one sees is that there are these small animals known as rotifers living quite happily. They're beating these coronal structures at the top of their heads to create currents to take food particles into their digestive system. And if we just zoom in on one of these guys and look at it by scanning electron microscopy, you can see the corona of cilia at the top, and then at the bottom you can see the foot that it uses to attach to the substrate in these colonies. Now the reason we're talking about these creatures today goes back to an observation that Peg Steyerwalt and Fred Lewis published 40 years ago, showing that rotifers living on the host snail can actually reduce cercarial output, prevent cercaria motility, and actually prevent and block infectivity as well. And so from this initial paper, what they described were 2 species of rotifers living on the snail shell, Rotaria rotatoria and Philodina acuticornis, which produced a water-soluble compound. One or both of these species produced a water-soluble compound that was capable of reducing cercaria motility. This was a really intriguing observation that I remember Fred telling me about when he was teaching us how to work with the parasites as we began our initial work on them and visited his laboratory. And so I thought this would be a really good project for a PI, me, to get back in the lab and try to make some progress. So as Fred was about to retire, I contacted him and asked him if he'd be okay with me pursuing this project, and he was kind enough to share his unpublished observations and also give me his blessing to pursue this compound, which they had been unable to actually completely characterize. So what I did was I went to the snails and removed from them these rotifers, and from individual rotifers then grew up clonal cultures of either Philodina acuticornis at the bottom or Rotaria rotatoria at the top. And from these clonal cultures then I could take the conditioned media and now add it to Cercaria and look at their motility. And in these movies I think you can quite clearly see that the Philodina conditioned water has no effect on this ability of Cercaria to move, whereas the Cercaria treated with Rotaria conditioned media are completely incapable of moving. And this is just shown here in these maximum intensity projections if we now take several of these frames and superpose them. So this basically said that 30 years after Fred's initial observation, these two rotifers were still living on the snail shells, and one of them was actually making a compound that was capable of paralyzing Cercaria. Now at this point, we wanted to know, how is it that Rotaria rotatoria is capable of paralyzing Cercaria? Or what is this schistosome paralysis factor? At this point, I was really lucky to have Jarong Gao join the lab as a graduate student, and here she is with our lab mascot, Schmitty. So to characterize this compound, we basically, as I told you, began with clonal cultures, clonally derived cultures, where we grew single rotifers up to expanded 1 liter cultures, lyophilized that media, subjected it to multiple rounds of molecular weight cutoff filtration, with the activity actually passing through the 650 Dalton filter, and then subjecting this to a first round of HPLC. And this work was done in collaboration with my colleague Jonathan Sweedler at the University of Illinois and his graduate student Ning Yang. So what the HPLC showed us is that in this red trace, which is the conditioned media from Rotaria, there's a peak at 25 minutes that's absent from the Philodina conditioned media, shown in blue. And that peak at 25 minutes is where this paralyzing bioactivity is located. Okay. So we now then subjected that peak to a second round of HPLC, determined its mass by mass spectrometry, and then grew up large quantities of the compound for NMR. And so for this, we used 25 liters of rotifer conditioned media, from which we obtained about 100 micrograms of SPF. And with that material, then, Jonathan and his colleagues were able to determine the structure of SPF, which is this novel tetracyclic alkaloid. Now, what you're looking at here is a dose response curve, just showing you the effects of increasing dosage of SPF on paralytic activity. So we're looking at percent of free-swimming larvae on the y-axis, and in red, there's no treatment with any compound. And then as we add increasing concentrations of SPF from 2.5 nanomolar in yellow up to 2.5 micromolar in purple, what you can see is that we eventually saturate and paralyze all of the parasites within a minute or so at higher concentrations. Now, this was a novel compound and one that has been thus far difficult to synthesize. But there were related compounds that had been described in the chemical literature. These two HT13 compounds, which were identified based on their affinity for human serotonin receptor. And if you look carefully, you'll see within all of these structures this serotonin backbone. Now, both HT13A and B had been synthesized, and they were shared with us by Bjorn Söderberg and Yanxing Jia. And what you can see is, although HT13B has no activity on Cercaria, the related compound HT13A is actually capable of paralyzing Cercaria at the highest doses that we tested. There were also related compounds that had been described during the course of these syntheses, and it turned out that this propyl derivative in the upper right was also capable of paralyzing Cercaria at concentrations equivalent to what we see with SPF, whereas these bulkier additions are basically wind up blocking activity. Okay, so with this information in hand about in vitro paralysis, we wanted to know whether this compound was actually capable of preventing schistosoma infection. What we can do is take known quantities of Cercaria, add them to a test tube with the drug, and now add the mouse tail to perform the infections the way we would normally in the laboratory. And now we can wait 6 weeks and ask, are there parasites? Can they be harvested from the host? And what does the liver look like? And so if we do this, what I'll show you is that if we treat the parasites with SPF, we can actually block infection and prevent pathology. Now, if we start with untreated Cercaria on the left, you can see that the mice infected with untreated Cercaria have swollen livers with lots of granulomas, and we recover lots of parasites from them. Similarly, at the lowest concentrations of these related analogs, again highlighted in pink here, we see, again, lots of granulomas, lots of parasites. By contrast, if we now infect with the lowest concentration, if we treat the parasites with the lowest concentration of SPF, 25 nanomolar at the upper left, what you can see is even the lowest concentration of SPF is already leading to reduced granuloma formation and reduced parasite infectivity. And we require about 2 orders of magnitude more of the related compounds to see similar effects. Now, as we increase the concentration of SPF even more, shown in yellow at the top, at 250 nanomolar and above, we fail to see granulomas in the livers and we block infection completely. And again, we require about 2 orders of magnitude more of the analogs to see similar kinds of effects. So in our brief time together today, I've told you about this intriguing chemical interaction across 4 different phyla. Biomphilaria glabrata is the intermediate host of the schistosome parasites. And living on this intermediate host are these commensal rotifers that produce this tetracyclic alkaloid that is capable of preventing these Cercaria from infecting their definitive host.
It appears that I'm already over my allotted time, so I'll just acknowledge my lab group. Here we are prior to the pandemic, post-pandemic, and I've already acknowledged the folks who've contributed to this work throughout the talk, and I will be happy to take any questions that you may have. Thank you.
Well, thank you very much, Professor Newmark, for these game-changing findings, actually. I do have a first question related into that, actually. It's 2 in 1. The first one was whether the addition of SPF and that effect, is it reversible? Is that something that can be cleared out once then you do remove that from the medium? Are they say carrier back then to motility? Or is that something that is irreversible? So lasting. And the second part of the question is also going in line with what Fiona Allen asks, which is whether these compounds, these particular ones, could have an effect on all the developmental stage of the parasite, actually.
Okay, thanks. So those are great questions. So if we wash out the compound, the effect is reversible. But if they're kept in it, the parasites continue not to move. But it can be washed out. So that would be the first part. And then in terms of effects on other parts of the other stages of the life cycle, we're still working on this hard. What we see, and these are preliminary, so this is amongst friends. We're still working on this. It seems that treating if Miricidia are treated with this compound in vitro and exposed to sporocyst transformation media, they fail to transform. So there is a possibility that this could also affect the ability of Miricidia to infect. But that's very much work still in progress. One of the things that's been difficult for us that we're still really struggling with is that we have not been able to synthesize this compound chemically. So it requires us purifying it from the rotifers to do all these assays. So we're working on that right now. try and figure out, both working with chemistry collaborators to synthesize it and also to figure out how the rotifer itself actually makes this compound. Because as many of you probably know, animals don't typically make these kinds of alkaloids. So it's very fascinating that the rotifer even has the machinery to do this.
Talking about the machinery to do this, have you tried also the rotifer's culture to expose them to antibiotic and seeing whether this product is still being synthesized?
That's great. That's a super important question. So that is a fantastic. So what the question is the rotifer making it itself, or is this actually coming from the rotifer's microbiome?
Yeah, totally.
So we are those are experiments that are ongoing right now. I have to say a lot of this work got slowed down almost completely by the pandemic. So we now have a new tech who's joined the lab, who's working on that very question, and he's been treating the rotifers with various antibiotics even as we speak. So hopefully next time I talk to you guys, we'll know the answer to that question.
Okay. And well, I have some questions which pretty much then from the audience will go in line with what you've mentioned. The first one is Giulio talking about the possibility of transferring then this laboratory result into an actionable solution that can then be applied then to the field. And I follow up then on this question by asking how stable is the compound then in the environment?
Yeah, so, you know, the compound does seem to be quite stable in that we can take the conditioned media and, you know, it certainly seems like the activity is retained reasonably well, certainly in the lab over time. In terms of, you know, I think the big question that we're all interested in, of course, is how can we actually use this for anything? And I guess our number one question about doing that, given that the related compounds that were identified previously were identified based on binding to human serotonin receptors, I think the one thing we have to be super careful about is what possible effects this could have on humans in the drinking water or in the water. And so I think that one of the major things that we have to figure out right now is how is this compound acting, and would it also be impacting the host in any way? And so that's really why we need to be able to make this thing and do toxicity testing before we even think about using it, I would say. But then I think, you know, there have been people have raised the issue of, well, is it worth putting rotifers into ponds in endemic areas? I think there's a lot of reasons to be cautious about doing those kinds of experiments. So I think I would actually love to talk to folks who would have much more experience in the field for ways that we could think about doing this. So I'm happy to have any discussion, anyone who wants to reach out and talk to me about this, I'd be happy to entertain those discussions from people who know a lot more about this than I do.
Oh, that's great. So we do have a lot of questions that are just pouring in. We might have to keep you for a little bit longer. So the next one is more into your arena. So developmental biology, is there a biological advantage of having these rotifers then producing or using the microbiota then instructing them to produce this specific compound? What do you think that will be for?
Yeah, so those, again, that's also just a great quote. Why is the rotifer making this? Is this beneficial to the snail in any way? All I can say at the moment is, so you could imagine that it could benefit the snail if it can block cercarial growth or motility or prevent further infection of the snail. It's hard to know how specific this particular interaction is. So one of the things I'd love to know is whether such rotifers are found on other species, on other populations of Biomphilaria, other than the one that's been cultured at the Biomedical Research Institute for the last 40 years. So it'd be really interesting to know in the field, are those rotifers out there showing similar commensal interactions, or are they different rotifers that don't make this compound? So that's something that we would love to know. At the moment, all I can do is speculate. You know, my gut feeling with no data is that the rotifer makes this compound probably as chemical warfare, probably to fight off other rotifers. Because I told you that there are these two rotifers that can live on the snail species, on these snail shells. And it turns out that the rotifer that makes this compound doesn't really grow very well on its own. so when we culture these things, it's the other species that doesn't make SPF that cultures really easily, that we can't kill, that is a great rotifer that can withstand everything. This Rotaria species is much less hardy. But yet when we take them off snail shells, Rotaria is the one that's there in more abundance. So my suspicion would be that actually this is chemical warfare targeting another rotifer and perhaps accidentally still hitting the same receptor molecules on schistosomes that it hits on these other rotifers. I have no data for that though, so that's just my model, and that's something as we figure out how this works, we'll hope to learn.
Definitely. So we're looking forward into this novel finding and continuity of the work. So thank you very much once more, then, Professor Newmark.
Thank you all, and I guess I'll look in the. I'll try to type answers into the Q&A if there's more.
Definitely,
questions.
there are quite a lot of them waiting for you there. So thanks
Thank
very
you all.
much.
Okay.
So we have reached now the end of our session, and I will hand over then the floor then to you, Debbie, so as to set then the directives for the break and the next part 2 of the session.